So, good morning, everybody. I know a few people are still logging in, but I'd like to welcome you to UAB Pathology Grand Rounds. Today we have the honor of hosting Carlos Orihuela, Orihuela from the Department of Microbiology here at UAB. Carlos started his training in Baylor and then received his PhD at UT Galveston. He then did his postdoctoral training at St. Jude before starting his faculty career at UT San Antonio. He was there for a number of years where he rose through associate professor before we were fortunate enough to recruit him to UAB in 2015. In 3 short years, he was promoted to full professor. And just last year, he became the director of our microbiome center. His research is primarily focused on host pathogen interactions responsible for the development of Streptococcus pneumonia disease, as well as other pneumonia-related issues. He has a very interesting and robust research program where he does some multi-omics or bioinformatics, where he combines a number of different techniques, including RNA sequencing and metabolomic profiling, as well as NMR mass spectroscopy. He's extremely well funded. He currently has 7 active grants, 4 of which are from the NIH as PI, 2 R01s and 2 R21s, as well as a prestigious HA fellowship, and then some collaborative grants. Along with that robust funding portfolio, he has over 100 manuscripts. He published 100 manuscripts, chapters, and a book that he edited. 8 of those were last year alone, including 1 in PNAS and Cell Reports. He puts his science and funding to good use. He's currently on 3 patents, one of which is for potential immunization on what he will be talking to us about today, which is acute and long-lasting cardiac damage as a result of pneumococcal pneumonia. So thank you, Dr. Orihuela.
Thank you, Adam. You can hear me?
Yes.
Great. Okay, so it's a pleasure to talk to you all today. This is an interesting project because, truthfully, it's something that I would never have envisioned I would have been working on. And yet it becomes obvious that someone should have been working on it quite a while ago. And so we just followed the science, and I think it's been very interesting. And I hope that I can at least get you to appreciate the impact of disseminated pneumococcal infection, and how when you get severe pneumococcal disease, the bacteria doesn't just stay in the lungs, or doesn't just stay in the pipes or the bloodstream. It really does get out, and it does considerable damage to all the other organs in the system. So I do have relevant financial disclosures. I have a patent on the YLN vaccine, and I'm going to talk to you a little bit about that. And I do receive licensing fees from Matrovax that's testing this in a clinical trial. So that's important. So the main actor in today's story is a bacteria called Streptococcus pneumonia. It's a gram-positive Diplococcus. You are probably most familiar with it as it's polysaccharide encapsulated, and that capsule is the basis of Prevnar, the vaccine that we give to children and older adults now. But what you need to know relevant to our conversation today is that this bacteria also elaborates a pore-forming toxin called pneumolysin. And this binds to cholesterol on the membrane of cells, oligomerizes, and forms pores that allow ions to come in and out of the cell and disrupts osmotic pressure if you have enough of it, and can even kill the cell. You need to know, obviously, that this bacteria is the leading cause of community-acquired pneumonia. And as a result of that, it is the leading cause of bacteremia and sepsis. And the risk groups are infants, elderly with underlying morbidities, those who are immunocompromised. And I should put on there individuals who have recently recovered from viral infection or are in the process of experiencing a viral infection of the respiratory tract. So this is what you would look up if you open a medical textbook. This is what you'll see generally regarding pneumococcal pathogenesis. The bacteria lives in the nasopharynx of humans, and it's spread by aerosol, maybe, but probably most likely fomites. Children passing it to children on toys, through saliva, and so forth. And it colonizes the nasopharynx, typically results in asymptomatic colonization, and then it just transmits to the next person and so forth. In young children, the bacteria can ascend to the eustachian tubes to cause otitis media. In older adults, it's aspirated to cause pneumonia. But in some cases, I think about 30% of the pneumonia cases, the bacteria is able to escape from the lung and enter the bloodstream. And in the bloodstream, the bacteria then has access to the other organs. And one of the most devastating consequences is meningitis. And to do that, the bacteria has to cross the blood-brain barrier, which is, of course, vascular endothelial cells that line the central nervous system. This is what's in the textbooks. This is what it was. This was there when I was a postdoc. In fact, I made this slide 20 years ago as a postdoc. So I did my postdoc at St. Jude Children's Research Hospital, and this figure pretty much summarizes the work. What we knew at the time was that the pneumococcus, which is Diplococcus, was binding to vascular endothelial cells in the bloodstream. So I was very interested in what was causing meningitis, how the bacteria caused meningitis. So that we are looking at this from bacteria in the bloodstream trying to get into the central nervous system. Okay. And so we knew that the bacteria bound to a receptor on endothelial cells called platelet activating factor receptor via phosphocholine moieties on its cell wall. We knew that the bacteria shed cell wall fragments and released this toxin pneumolysin. This caused inflammation. This caused cell death. So the blood-brain barrier becomes a little bit leaky. It becomes inflamed. And these cause the upregulation of platelet activating factor receptor and an unknown ligand that we knew the bacteria bound to via a protein called CBPA. So these two interactions, CBPA interacting with something and phosphocholine binding with PAF receptor, were critical for bacterial translocation across the blood-brain barrier to cause meningitis. And my job as a postdoc was to identify that question mark, identify the ligand for CBPA. So the way we approached this a long time ago was to use cranial windows. And what we do in this case is we actually remove the top part of the skull of the mouse and we replace it with a glass cover slip. So this is a mouse, the top of the head. You can see that I can look straight down onto the brain. I can see the vasculature. You know, we've removed basically the dura mater, and we're looking right down onto the subarachnoid space. And you know, this is sealed, so this animal walks around, does everything you can, but you can also put this mouse under a microscope, and as a result, you can go and look at the vasculature that is going into the brain. And so these are blood vessels that are on top of the brain going into the actual neurons, and these blood vessels are surrounded by cerebrospinal fluid. So what we would do is we would inject fluorescent latex beads into the mouse, and we can coat these beads with different proteins. And so, for example, here, these latex beads are coated with choline binding protein A, recombinant protein. And you can see that within a few seconds, these beads are binding to the vasculature in the brain. And this is very interesting. It's a very powerful tool because we can modify choline binding protein A, we can make mutants in choline binding protein A, we can load it up onto these beads, and then we can see what is the effect of binding on the vasculature. And this is relevant because, one, CBPA binds to something on vascular endothelial cells versus control. So again, whatever it's binding to is that question mark factor that we were trying to identify. And two, we knew that if we left the beads in the animal for a while, the beads translocated. So here you can see 10 minutes after challenge, the beads are bound to the vasculature. And yet, come back a day later, the beads are now on the opposite side. They're now in the central nervous system. And you can see that clearly over here, where the beads have extravasated across the vascular endothelial cell. So this was a powerful model for us to use and to dissect what CBPA might be doing, and hopefully also to validate what CBPA might be binding to if we find any targets. And so we actually, I'm not going to show you all that data, that's all published, but we did a series of pull down experiments, we did a series of studies, and we identified the 3767 kilodalton laminin receptor as the ligand for choline binding protein A. And that is a really interesting protein because, as it turns out, there are many, many other pathogens that bind to this laminin receptor, and they all tend to be neurotropic. Prions bind to laminin receptor, and that is the basis of them entering the central nervous system. VE virus, some E. coli toxins, Symbis virus, those are both cause neurological problems. Interestingly enough, syphilis has recently been discovered to also bind to laminin receptor, and that's thought to be an important part of its neurotropism as well. So laminin receptor seems to be a chink in the armor, so to speak, of the blood-brain barrier that allows the bacteria to attach. You can see here that if we give beads, CBPA-coated beads, but immediately prior to that, administer anti-laminin receptor antibodies, we drastically reduce the amount of beads that attach to the vasculature in our cranial window model. And here is basically in vitro experiments where we, these are all where we looked at the ability of bacteria to cross vascular endothelial cells. And you can see that here normalized to 100%. If you have 2 antibodies against laminin receptor, you are able to reduce adhesion by 5-fold. If you treat the cells with laminin, which is the ligand for laminin receptor, that apparently sterically blocks bacterial binding as well. And if you have a bacteria that lacks CBPA in its entirety, it doesn't bind to the cells at all. And of course, it doesn't matter then if you have these treatments because it can't attach to the vascular endothelial cells anyways. Okay, so that was pretty interesting, and we published that, and this will become important later. But one of the observations that we had at the time was that if we injected intact bacteria or we injected cell wall fragments that contain choline binding protein A, is that we saw a dramatic drop in blood pressure and the speed which the beads flowed through the heart, okay? And what we eventually learned is that bacterial cell wall was inhibitory of cardiac contractility. So this was just an observation that we made at the time. And so this is left ventricular pressure of rat hearts that have been isolated. And when you treat those hearts with a cell wall, you can see that the left ventricular pressure drops. And if you don't treat them, it stays high. If you treat them with a version of ethanolamine, a version of cell wall that does not have a residue called phosphocholine on its surface, which again binds to the vascular cells, but is also the anchor of CBPA, which is short for choline binding protein A, that's sustained. And if you give a PAC receptor antagonist here, you protect. So the main thing here is that in those studies to look at how the bacteria crosses the blood-brain barrier, we discovered that there was an impact of the bacteria on cardiac contractility, and it was suppressive. And this might have been contributing to some of the mortality that you see during a severe infection. Now, at the same time that we made that observation, Dan Musher, an infectious disease doctor at Baylor College of Medicine, and the VA in Houston, he made this report. He published this report. And basically what he did is he looked at the incidence of adverse cardiac events following hospitalization in patients diagnosed with confirmed pneumococcal pneumonia. And he knew, or he showed in this paper, that about 20%, or 33 out of 107 individuals in the hospital for pneumococcal pneumonia experienced some form of adverse cardiac event. And that was myocardial infarction, arrhythmia, or congestive heart failure. Most typically, it seemed to be a combination of these three different types of adverse cardiac events. However, the most interesting one is that the majority of these were congestive heart failure. Okay, and that's interesting because it's not myocardial infarction, and by what we really are saying here is there seems to be something happening to the heart versus an occlusion or a blocking of a blood vessel that then impacts the heart. This is more of a functional property of the heart itself. One of his key results was that patients with concurrent pneumococcal pneumonia and cardiac events had significantly higher mortality, approximately fourfold than those with pneumonia alone, and that was significant. So the people that were dying at the VA as a result of pneumococcal pneumonia were actually the ones that were having pneumococcal pneumonia and some form of adverse cardiac event. So obviously, this observation, along with our own, that pneumococcal cell wall products are inhibitory of cardiac contractility, was very interesting, and that seemed to be like an area that we'd want to investigate further. This is now several years further. I'm just going to show you a little bit of data that kind of highlights this. This is a study by Ulrich et al. And what they did is they looked at what are the risk factors for adverse cardiac events following pneumonia. And they looked at all of the possibilities, everything that might have happened while a person was in the hospital. And as you can see very clearly, there's one factor that stands out way above all the others, and that is pneumococcal bacteremia. And very interestingly, of course, is that non-pneumococcal bacteremia does not. So there's something special about the pneumococcus that seems to damage the heart and put a person at risk for heart failure after they've been released during convalescence. And this is for up to 10 years following. So this is what we discovered as sort of being responsible. And it's astonishing to me that no one had reported this. I've asked, and people had seen these, they just weren't sure what to do with them. And it's simply the idea that the bacteria gets into the heart muscle and is able to replicate and kill cardiomyocytes directly. These are mouse hearts, but you can see here we've highlighted foci of infection, and upon closer examination of the hearts, you can see that there is pericarditis. The pericarditis is typical. There's a lot of immune infiltration here, but interestingly, adjacent to blood vessels, you see these little foci. This one's next to the little capillaries, and these foci seem to be filled with bacteria. This here is a magnification of some of those images. And it's just astonishing, right? You can see that the bacteria are in the heart, they're replicating, there's lots of them, and there's no immune cells, which is striking, right? It's very different than the pericarditis that you would have seen before, and it's different than staphylococcus. Staphylococcal abscesses are filled with neutrophils, PMNs, monocytes. This is not the case for these lesions caused by strep pneumo. And of course, again, you can see that they're typically, it looks like they kind of start next to a blood vessel and then they just sort of spread and take off. We've gotten more sophisticated in our ability to identify the lesions. Now we use fluorescent microscopy. This is a heart again of a mouse, but now we stain for capsular polysaccharide. So this is the serotype 4 isolate, and we can see fluorescently the lesions. This allows us to do quantitative analysis using FlowJo. So now we've taken out of its unbiased measurements of bacteria in the heart. We've also gotten the ability to do EKGs on mice. We also have echocardiograph. I'll show you that a little bit later. But one of the things that's very obvious is the more lesions the mouse has, the more likely that it's going to have aberrant cardiac function, right? And this is the same animal, pre-infection, 30 hours later, and the heart. And this is what a typical heart would look like for an animal at 30 hours. It's not the same animal. So these lesions seem to be disrupting contractility and the basic electrophysiology of the heart. When we look at the lesions and we start doing detailed microscopy, one of the things that we see is that there are dead host cells inside of the lesions. That's to be expected, right? If the bacteria are replicating, you'd be sort of surprised if there weren't dead cells. And these are tunnel positive cells, so this means these are cells that have died in some fashion. We detect the bacterial toxin pneumolysin where the lesions are. Okay, that's important because now we have a direct cytotoxic factor at site. And very interestingly here, sorry, I have a picture, you guys have to move it every time I'm talking. Here, you can see these are troponin levels in serum of animals infected with the bacteria. And you can see that in the mock, we don't detect any troponin. In an animal infected with D39, this is a serotype 2 strain of pneumococcus, we see considerable levels of troponin in the serum. But if we have pneumolysin deficient mutants, these guys, even though they're able to get into the heart, they don't form the lesions and they don't cause the release of troponin. So this cardiac damage seems to be directly dependent on the bacteria's ability to get into the heart, but then also to elaborate the toxin pneumolysin that is cytotoxic. Here, I sort of show you what lesion development looks like over the course of time. We know at the very, very early time points that it's actually individual bacteria that are able to cross into the heart. And as time develops, as time moves on, these individual bacteria are, I imagine some of them are cleared by resident macrophages, but some are not. And those that don't are able to establish a foci of replication, and over the course of time, they replicate and so forth. There is a moment of time when there is immune cell infiltration, and these are typically when they're smaller. But for reasons that are not entirely clear, these cells go away. They're either killed, they're probably killed is our thoughts. And they can come back sometimes, but they don't ever make up the majority of the lesion like we would be expecting for a staphylococcal abscess. It's important to remember this is not happening in a vacuum where you only have a heart that's infected. These animals have pneumonia. These animals have bacteremia. They are septic or borderline septic. And then there's also bacteria in the heart. Okay, so one of the reasons there might not be such a strong recruitment of immune cells is that there are chemokines in every other organ of the body calling for immune cells. And it's possible that the gradients are just stronger in other organs than they are for the heart. So that the immune cells get pulled to the liver, to the spleen, to the lungs, versus being recruited into the heart. So this is not happening in isolation. This is happening in the background of a severe disseminated infection. This is electron microscopy of an infected heart, and it just goes back to what we were saying about the bacteria seem to attach and translocate through the capillary bed of the organ. And then it seems like they are able to establish a foothold and replicate. We actually strongly suspect that this is what remains of a resident macrophage, and that this macrophage probably consumed, took up the bacteria, and in this case, the macrophage was killed, and the bacteria is able to then use that dead macrophage as a niche to replicate. So when these animals are bacteremic, they have anywhere from 10 to the 5th to 10 to the 6th bacteria per ml. In a typical mouse heart, we probably see at endpoint, we probably see 20 to 30 lesions, okay? So this is actually a very rare event given the number of bacteria constantly streaming through the heart of this animal and the duration of time. So what probably happens is the macrophage wins most of the time and nothing happens. But eventually, in some cases, the bacteria win. And that's enough to allow this process to start and to move and to happen. Here, we started to get a little bit more molecular. And so these are blood vessels in a heart. And what we're doing now is we're staining for platelet activating factor receptor. That's one of the ligands the bacteria uses to attach and cross vascular endothelial cells. And here we're staining for laminin receptor. That's the other ligand. This is the one that CDPA binds to. And you can see that one, they overlap, but two, they overlap with the stain for vascular endothelial cells, which is tomatolectin. So these two receptors that the bacteria uses to cross the blood-brain barrier are actually the same receptors the bacteria uses to cross the cardiac vasculature and gain access to the myocardium. So here's our blood-brain barrier model again, but what we've done now is we've applied this to context of the cardiac vasculature, looking for development of lesions and looking for their ability to grow in size. And so here is, we'll start with the number of lesions. Wild type, you can see at 24 hours, we see about 5 to 10 in an animal. And then by 30 hours, there's around 25 to 30. With the CDPA knockout, this bacteria can't cross vascular endothelial cells, right? This number is strongly diminished. And with the pneumolysin knockout, you do see some lesions at 30 hours, but it's still reduced versus the wild type. Importantly, in the pneumolysin mutant, the lesion size is dramatically reduced. So even though there's some lesions here, these lesions are smaller in size, and that has to do with the fact that it appears killing the host cells seems to be a vital part of the process of lesion development. If we treat animals with anti-laminin receptor antibody, which we showed you blocks translocation in vitro, we protect against lesion development in vivo. And if we use platelet activating factor receptor knockout animals, we also are able to protect the animals from development of cardiac lesions. So again, the bacteria needs these two signals to be able to cross, and just blocking one seems to be sufficient. And then once it gets in, apparently it needs pneumolysin to be able to do what it wants. So that leads to YLN. So this is a vaccine component that we developed in collaboration with St. Jude. And what it consists is, this is one of the domains of choline binding protein A. There are two motifs on the R domain of this protein. One is this NEEC loop, and one is this YPT loop. The NEEC loop is what's responsible for binding to laminin receptor, and the YPT motif is important for CDPA binding to actually polymeric immunoglobulin receptor. And this allows the bacteria to attach and invade endothelial cells and epithelial cells. This is just a pneumolysin toxoid, so it's pneumolysin. It has a single point mutation that stops this toxin from oligomerizing and therefore forming cytolytic pores. Okay, and then we've created versions where we take YPT or NEEC and we add these at the end, and a version we call YLN where we have both versions at the front and on the back. These are cardiac lesions in animals that were immunized with all of these different constructs. Here's alum as our control. And you can see that CDPA confers some protection, L460D, the pneumolysin toxoid confers some protection, but the most robust and significant in this study are in fact the versions that have a combination of pneumolysin and the NEEC domain, which is the one that binds laminin receptor. So we were able to protect against cardiac lesion formation in this model using this antibody, this fusion construct. And in separate studies that I'm not showing you, this same vaccine hybrid is able to protect against development of meningitis. Again, the same mechanisms are at play for crossing the blood-brain barrier as they are for crossing into the heart, and most likely crossing into other organs, as this seems to be shared, a common thing for vascular endothelial cells. Okay, so that means that we have advanced our model, and now we're starting to think, I think, in broader terms. Everything I've told you is the same, except what we're really saying here now is that during bacteremia, there seems to be the idea that the bacteria stays in the pipes, again, stays in the bloodstream and doesn't really have any consequence other than being in the bloodstream and maybe causing sepsis is wrong. The bacteria is actually getting into the organs, and in there it's damaging the organs. And the one we've looked at specifically is the heart, and of course that's what this talk is about. But it's foolish to think it's only the heart, right? And if we look at the kidneys, if we look at the spleen architecture, if we look at other organs, we actually see that the bacteria are in these other organs as well, in the parenchyma. So this bacteria is everywhere, and it is damaging. Now, this is where it continues to get even more interesting. So this is a mouse heart 7 days after being successfully treated for invasive pneumococcal disease with antimicrobials. So this animal has no bacteria in it at all. We've successfully treated it, and it looks otherwise healthy, at least when you look at it at a case. Importantly, animals that are intervened that way, at day 0, we don't really see anything in the heart. At day 3, we don't see anything. But by day 7, you start seeing serious cardiac remodeling, primarily in the deposition of collagen around areas where the microlesions used to be. So it seems that, and I don't show it here, but these areas have been infiltrated by monocytes. So what's happened here is that the heart has now entered its repair mode, and it's now trying to heal itself. And this is resulting in collagen deposition and cardiac remodeling. This is very compelling because, as I indicated earlier, one of the things that we know is that if you've been hospitalized for community-acquired pneumonia or severe pneumonia, you're at risk for adverse cardiac events for many, many years after the fact. This is one study here where they look at the adjusted hazard ratio of individuals hospitalized for pneumonia. So these people are convalescents. And while the incidence of adverse cardiac events is highest in the first 3 months, it never quite goes away. Right here, 5 years later, you're still twice as likely as the control groups are for having an adverse cardiac event after hospitalization for pneumonia. This study is very powerful. In this study, they measured levels of cardiac troponin in individuals hospitalized for pneumonia at time of admission. And then they looked to see how long these individuals tended to survive. And you can see that in the lowest tertile, those that had troponin levels less than 14, almost all these individuals were alive 4 years later, whereas in the group that had the highest levels of troponin upon admission for community-acquired pneumonia, less than 30% were alive 4 years later. And again, and not in this study, but when you start diving into the literature to see what is it that these individuals tend to die of, adverse cardiac events is one of the most prominent causes of death. This is a paper that came out about 3 years ago, and it sort of highlights the idea that there is a tremendous amount of systemic damage following community-acquired pneumonia, or at least severe community-acquired pneumonia that requires hospitalization. So this is the age of, I guess, 154 people at the time that they were discharged. Some were very old, some are quite young, right? So this is how old they were when they were discharged. And based on census data, this is how many years they are expected to live beyond the time that they were released. So a 100-year-old person might only live 1 year, and you would expect that a 25-year-old would live another 40, 50 years. What you're looking at here, these bars, are actually how long they lived before they died. And one of the clear things that comes up is that there's a lot of empty space between expected lifespan for individuals and actual lifespan of individuals. And this suggests that hospitalization for community-acquired pneumonia is actually cutting into expected lifespan. And there are many studies now that show that if you've been hospitalized for community-acquired pneumonia, you're likely to become more frail, you have cognitive declines, you're likely to lose independence as a result of this, and obviously this paper shows you're also less likely to live your full life expectancy. And we suspect that this is because of this disseminated organ damage that is occurring during these severe bacterial infections. Importantly, everything I've showed you is in mice. Mice are not men. And so one of the key things that we tried to do and we were able to do is to do studies in non-human primates. This was done in San Antonio. These baboons were given pneumococcal disease. They had pneumonia. We looked at the hearts of these animals. We were able to detect bacteria. We were able to see considerable cardiomyopathy. And we were able to see strikingly very high levels of collagen deposition in convalescent animals when they were treated with antibiotics. It's important to note that these primates were not as sick as the mice. Because of ethical reasons, we had to stop the experiment at the point that the primate would have been intubated had it been a human, right? So we had no ICU equivalent for the primate. So there are people who actually become sicker than these primates were at the time that we intervened, where we sacrificed animals. So it's possible these are probably on their way to becoming these lesions that we've described. So at this point, I want to summarize. Severe pneumococcal pneumonia is associated with adverse cardiac events during hospitalization convalescence, and these negatively impact mortality. During bacteremia, the pneumococcus is able to invade the myocardium, replicate, and directly damage the heart, and this suppresses contractility. Pneumococcal translocation is via the same mechanisms as blood-brain barrier translocation, and this can be blocked with antibody against the wild construct. And pneumococcus kills cardiomyocytes in a pneumolysin-dependent manner. Okay, I'm quickly going to change gears. This is another part of our lab's research effort. This is a cell death pathway called necroptosis. It's typically triggered as a result of TNF receptor engagement in absence of caspase-8 activation. This is a way that cells die when apoptosis is blocked. Okay, and it's really important for viral infections because viruses block apoptosis and the cell has to have a way to still get rid of itself in case that happens. What you need to know, or what's relevant, is that pneumolysin can activate this pathway independently of TNF receptor engagement, and it does it by the ion dysregulation, ATP depletion, and somehow that activates this RIP1, and then that leads to the development of necroptosis. So one of our goals was to determine if the cardiac damage is long-lasting, that scarring that we see in the mice and the primate, is it real? Does it last? Can it really explain what happens 10 years down the road? Does pneumolysin kill the cardiomyocytes via necroptosis? And can necroptosis be blocked? Is maybe this a way to protect against disease? So in this slide, what I show you is that cardiomyocytes in vitro, at least, they die of necroptosis. Phospho-MLKL, that's this protein right here, is the end product of this necroptosis pathway. So if we are able to detect phospho-MLKL, we know that necroptosis is occurring. And here you can see that cardiomyocytes treated with bacteria, there's lots of phospho-MLKL. And if these bacteria are treated with the pneumolysin deficient mutant of the bacteria, there's no phospho-MLKL. So the cardiomyocytes infected in vitro at least, are undergoing necroptosis. If we block necroptosis in vitro, and we can do this by a variety of pharmacological agents, NEC5, GSK, and NSA, none of which of these are possible for humans, but they do work in mice. We are able to reduce cytotoxicity. We're able to prevent cells from dying. This is actually siRNA. We're able to protect cardiomyocytes in vitro against bacterial-induced cell death. In vivo, at sites of cardiac lesions, we are able to detect phospho-MOKL where the bacteria are present in these mouse hearts. And importantly, if we take necroptosis deficient animals, those that are RIP3 or MOKL deficient, these are the signaling pathway, and this is the effector again, you can see that we see a reduction in the amount of troponin that's present in the bloodstream of these animals. And we also see a stark reduction in the number of lesions in tissue sections. So necroptosis or cell death, again, seems to be an important part of the formation of these lesions. We go back to the idea that maybe these cells are sort of like an intracellular reservoir for the bacteria. It's a little niche that the bacteria can sort of establish their footprint in the heart, and then things sort of take off from there. So cell death is an important part of that process. This is sort of the same thing I've showed you, but the difference is, in this case, we infected the mice, we treated them with ampicillin, and then we went 2 weeks, 1 month, and 3 months post-intervention to look to see if cardiac remodeling was still present. In those last slides I showed you, all of that was done at 7 days. So in this case, now we've gone further. And what I can show you is that the amount of collagen that we detect in the heart, first of all, it's always greater in the severely infected animals versus the mildly infected animals. And our cutoff for this was bacterial titers in the bloodstream of above 10 to the 5th. And it seemed to be long lasting. So there was detectable collagen in severely infected animals and even mildly infected animals that was greater than the uninfected control as far as 3 months out. We started to do echocardiography with the resources here at UAB. So here you can see that the ejection fraction and fractional shortening of mice that have been infected 3 months earlier and rescued, that it's impaired. And importantly, what you see is that the heart does contract, but whereas the heart is supposed to contract evenly towards the middle, these green lines represent the vectors where the heart cell, the heart wall is contracting towards. In a mouse that's infected, you lose that focal point where the heart is contracting. So the heart is contracting, but it's contracting unevenly, or well, unevenly, I guess is the best word. And that's really evident here where we look at segmental synchronicity. This is the walls all coming together at the same time. And you can see it's very much disrupted in an infected animal, even 3 months after. And so this helps to explain why the ejection fraction is altered. So in 2015, other investigators looked to identify inhibitors of necroptosis. They weren't interested in pneumococcal infection. They were doing it for other purposes. And they discovered that 2 drugs, which are used to treat forms of leukemia, resistant versions of leukemia that don't respond to normal tyrosine kinase inhibitors, that there were 2 drugs, tizapinib and ponatinib, that actually blocked the necroptosis pathway. And ponatinib, which is the drug we're going to talk about, actually blocks it at 2 spots. It blocks RIP1 activation and it blocks RIP3 activation. And in the paper that they identified these drugs, they say, these FDA-approved drugs, blah, 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 are potentially interesting for the treatment of pathologies caused by aggravated necroptotic death. And we agreed with that. And we said, what if we give necroptosis inhibitors to our animals? So the first thing is that blocking of necroptosis changes the characteristics of the heart lesion. Here we have this heart lesion, and you can see it's filled with bacteria. There's a few immune cells around it, but there's nothing in the middle. And when we give the necroptosis inhibitor, in this case, necrostatin 5, not ponatinib, but I'll show you that in a second, you now see that there are PMNs and neutrophils all over the lesion. Okay, and the amount of troponin and the number of lesions per size decreases in ponatinib-treated animals. And these animals were not pre-treated. I want to point out all our experiments, the drug is given therapeutically at the same time that the animals receive antimicrobials. Okay, so typically in our IP model, not our pneumonia model, but in our IP model, that is 1 day after challenge. These mice are bacteremic at that point. And if they don't get antibiotics at that point, they will die. Or even if we wait longer and they get antibiotics, they'll still die. So it's kind of like the end point that we can, as far as we can push the animals and still be able to bring a reasonable number of them back. So this is adjunct therapy to antimicrobials. Importantly, when we look at collagen deposition in ponatinib animals, now you can see that the amount of collagen deposition is dramatically decreased at two weeks, at 1 month. We sort of lose resolution at 3 months, but if we had been able to have more animals, we probably would have been able to make this significant. If we look at contractility, we're able to not prevent, but able to diminish the loss in cardiac contractility. So fractional shortening is a little bit better, and ejection fraction is a little bit better in ponatinib-treated animals versus vehicle. So these animals were sick. These guys were sick but got the drug. And you can see that the ponatinib-treated mouse, the segmental synchronicity is a little bit better than the animal that was infected but wasn't sick, but not as good as the uninfected control. So ponatinib is blocking the prevention, is not preventing lesion formation, but changing how the host responds to the lesion after the fact and reducing the amount of collagen deposition and the amount of long-term, or at least for 3 months, cardiac damage seen in these mice. I just want to conclude and tell you that necroptosis is happening in the other organs that are affected by pneumococcus. After all, pneumolysin is released everywhere the pneumococcus is. These are our non-human primates again. And all that really matters is that necroptosis is happening in the airway. And depending on how sick the animal is, you see more necroptosis, right? So that makes perfect sense. The more bacteria, the more pneumonia, the more necroptosis. Necroptosis also seems to have an important role during influenza pneumococcal co-infection. We just published a paper that shows influenza primes lung epithelial cells for pneumolysin-mediated necroptosis. It's important to remember that necroptosis is a defense pathway to kill cells that are infected by viruses and can't undergo apoptosis. So now you have cells that are infected with virus and they're being exposed to pneumolysin. So maybe there's a double signal there. It gets amplified, but whatever happens, you can see that the amount of necroptosis in virus, this is influenza A virus and this is pneumococcus, is drastically more pronounced in these co-infected animals. And we can protect the airway from damage with ponatinib as well. If you look closely though, these lungs are not normal. There's actually quite a bit of immune cell infiltration in these lungs. So we need to look into that because we're protecting the architecture of the lungs, but we might actually be causing too much of a robust immune cell infiltration. And this is one of the areas that we're studying. Okay, so these are the same conclusions, but now I'm adding just a little bit more. Pneumolysin-mediated cell death is a result of necroptosis. Cardiac damage is long-lasting, and this potentially explains the adverse cardiac events in convalescence. And drugs that block necroptosis may be a viable adjunct therapy for pneumococcal disease in general. Okay, so this is the lab. We have a lot of support from collaborators across UAB and at other sites. Obviously we do okay with funding as well. So thank you. I'm happy to take any questions.
That was great. Thank you. It looks like we do have at least a few questions from Dr. Davis. If do you want to unmute and ask them, or do you want me to read them?
Whatever's easier for you.
Seeing if he was still there.
Yeah, I'm still here. I can
There he
ask.
is, yeah.
That was excellent. I'm a forensic pathologist, so I don't always get to hear presentations about death. That was great. I've had three questions. I couldn't help but think. You're one, the study of decreased life expectancy in general in that 30-year study. I think the youngest persons were about 25 or so. So I take it no one has studied this in children. But do you have any thoughts on whether this would hold true for a 2-year-old?
We're really interested in that, and we wrote a grant with a collaborator in Peru to try to do that because honestly, pneumococcal pneumonia in children doesn't really happen anymore here in the US. The vaccine's quite good. We are very interested. We don't have the answer to that.
Okay.
What I can tell you is that children with sickle cell disease who are very subject to invasive pneumococcal disease. St. Jude now does EKGs and monitors contractility in them as a result of this study, of this work. And they are seeing aberrations in cardiac function in the children with sickle cell disease who end up with an invasive infection.
All right. And then I just wondered if various communities who would be interested in this know about it. One would be the public health community. The other would be the life insurance industry.
Well, maybe I will keep that second one quiet.
Okay.
But no, I mean, you're right. I mean, it's very intriguing, right?
Yeah.
I think it comes down to, so I've gone back and I've read the pre-antibiotic era literature. And believe it or not, this is described. It's in there.
Oh, I believe that.
Right? They talk about it, and in fact, they used to use EKGs as sort of a way to figure out if there was exactly what you were experiencing. I think it got to the point where we simply said we can eradicate these things with antimicrobials, and we just sort of forgot that, you know, basically severe pneumonia, invasive disease, is a train wreck, right? And you can put those trains, straighten them back out, put them back on the tracks, I guess, but it's not going to be brand new again, right? You've damaged the organs, and there's going to be consequences to those in the long term.
Okay. Well, thank you. Excellent work. I might have to go back and look at our own cases here in the office.
Please do, and if you find anything, let me know.
All right. Thanks.
Raj, do you have a question?
Oh, yes. So, Dr. Leal, that was an awesome story and very, very impressive data. So, I am wondering, like in any of your models wherein like acute versus chronic infections, did you ever see a pattern where you see a preserved ejection fraction and, you know, adaptive remodeling in the beginning and leading to associated with sort of diastolic dysfunction?
So pneumococcal pneumonia really only causes acute pneumonia. It's not really a pathogen to cause chronic disease. I think in a typical human, you know, from the time that you start coughing to the time you might need to be hospitalized for pneumonia is less than a week, probably more like 3 or 4 days. So everything happens very quickly with pneumococcal disease. There are other pathogens apparently that also cause these type of lesions, and that's been published now. One of the more interesting ones is Mycobacterium avium. There's published reports now that forms cardiac lesions too. Another one is enterococci. That's interesting as well, because that might be a way it damages the heart in addition to infective endocarditis. And then other invasive disease, Francisella tularensis and Francisella novacida also cause these lesions. So pneumo doesn't cause chronic infections, so I can't really comment to that. And I'm not a physician myself, so I've never actually treated a patient with this. I can't tell you anything clinical. But apparently this is a little bit more widespread than just pneumo.
Thank you.
So one question that comes to mind is, and this probably has more to do with the last part of your talk, with COVID, there's been a lot of talk about the myocarditis in particularly college athletes that are otherwise healthy. Do you think some of the TKI therapies may be warranted?
No.
Okay.
No, because I think that what's happening with those inhibitors is they are blocking the cell death pathway, preventing cells from dying, and maybe some of them have a chance to recover, but necroptosis is an inflammatory mode of cell death, right? So it's going to aggravate local tissue damage. Versus what's most likely happening with COVID is you have a runaway inflammatory response, right? That's leading to the fibrosis, we assume, right? So those are different sort of mechanisms of why you would have collagen depositions, right? It would be interesting if you find a treatment to protect against COVID collagen remodeling, that might be applicable for pneumo, right? After the fact. But I don't think the other way around.
Okay. Are there any other questions? It was an excellent talk. Very, I like how you brought it through the models, and we really don't see much non-human primate work, so very applicable.
Yeah, just to tell you why I know I'm not a physician, I never stepped into the room with the primates even. That was too close to home even at that level. So we were very fortunate to have outstanding staff to take care of that for us. Yeah.
Cool. Well, thank you so much. That was excellent. Definitely hit the spot, and it's great to hear that such wonderful work's going on. Hopefully that vaccine gets out there soon and widespread for you as well. As far as the patching goes. So thank you all for joining. Hopefully you can join us next week. We're going to have an external speaker, Jennifer Hunt from University of Florida, and we're going to be changing gears, and she's going to talk about reversing the gender leadership gap, starting from the inside out. So quite a bit different from the last two talks, but I think also very important and timely topic. So hope to see you all next Thursday. Have a great day. Thank you, Carlos. It was wonderful.
Thank you.